So I am the Region 3 Aquatic Invasive Species Coordinator. So my region overlaps a bit with CRISP as well as Lower Hudson Prism. So this is work that I have done, and then also Steve Pearson, who is the research scientist for the DEC, has done on northern snakehead in and around kind of the CRISP Prism region, especially in, as was mentioned, the Delaware Basin and then the Bashak as well. So as an introduction for anybody who, doesn't know, northern snakehead is this really interesting looking fish that's native to most of Asia, was introduced into the United States at several different locations. It was likely that these introductions were kind of a mixture of aquarium dumps of unwanted fish, as well as releases for cultural reasons, and people really like to sport fish northern snakehead, and so there have been some individuals who have released snakehead into the water with the intention of creating a population for sport fishing reasons. And then once they're in there, we've seen that they're extremely prolific at being able to reproduce in these introduced systems. Some of their ecology is they're a freshwater fish, although they're fairly salt tolerant, being able to tolerate up to 18 parts per 1,000. They like to exist in sort of stagnant, shallow water with muddy substrate, and so sort of swampy, marshy areas is where they end up. And then they can use rivers like the Delaware and like the Bashak, and hopefully not like the Hudson, to travel up and down and get into new areas. They also tend to hang out in areas that have a lot of aquatic vegetation. And again, so it's, these areas that we tend to not like to go, where you're going to sink in mud up to your waist, is where they like to hang out. Seasonally, they head out to deeper water in the winter. That way they don't freeze, but that means that they are able to overwinter, even in colder climates like this one, if they can find deep enough water so they don't freeze. In the spring and fall, they'll head back out to those shallow vegetated areas, and we've seen that they have an upstream dispersal of around 18 kilometers. So they can move pretty far in a season, which is why we've been doing our best to kind of keep track of where they are and where we expect them to be within the next season or two. They'll eat pretty much anything that will fit inside their mouth. That means when they're small, it's zooplankton, insects, and crustaceans. When they're, juveniles, they'll eat small fish, and then adults will eat fish up to 1/3 of its length. And these fish can get fairly sizable, and so, they can predate most of the other fish within a system. Within their native range, we've seen that within 2 years, they reach sexual maturity, and then are able to, at that point, spawn up to over 100,000 eggs per year. They spawn anywhere from 1 to 5 times per year, and they exhibit parental care. And so these fish have guarding instincts, and so you'll see fry ball, and then an adult is usually accompanying that fry ball. And they can live up to 8 years in the wild. So these are quite a long-lived fish. And unfortunately, we've seen from some studies in the Potomac that in their, expanded range habitat, they actually reach sexual maturity within about a year, so a little bit sooner, which gives them even more years to reproduce in their expanded range, which is not good news for us. So in New York and the Northeast, where they are most prolifically at this point is in the Susquehanna and the Delaware River. If we zoom out a little bit to a wider scale, there are, a lot of places kind of in our mid-Atlantic region where they are at. We have New Jersey, Pennsylvania, New York, Maryland. And then there are also some populations in Georgia and Florida, and some other individual populations kind of scattered around the US right now. Again, because of how they're introduced, which is human introductions, like aquarium dumps, it's not uncommon to see these kinds of little, point introductions that then expand out, kind of like what we're seeing in our area right now. So in New York, the earliest introduction that we've seen in the state was 2005 in Queens. We know that population is there and it's being monitored. And then we saw a population in Orange County. This population was introduced into an enclosed water body, so there wasn't, it was possible for us to eradicate that species using a pesticide. But, when we did that treatment, we were really able to see how quickly they were able to reproduce since we had detected them in 2008. Overall, in the entire treatment, throughout the entire eradication, we got 250 individuals that were taken out in that eradication. So they are very prolific and they can become a problem very quickly, we've seen. Unfortunately, that isn't the chemical treatment we were able to do in Orange County isn't exactly realistic for these larger riverine systems like the Bashakill and like the Susquehanna and the Delaware. So in the Delaware Basin, in 2019, we got a detection in the river, and then in the Bashakill WMA, we got a detection in 2021, as well as some unconfirmed reports in the Oswego and the Hudson. And we do occasionally get some unconfirmed reports in of people catching what they think is a snakehead, and often that turns out to be a bowfin, or it's just not a correct detection. But we have to take those reports fairly seriously. And how we do that is for our snakehead sampling and early detection monitoring system. We do tandem sampling of eDNA and electrofishing. So if an angler says they catch a snakehead, in this case, maybe they don't provide a photo, our inland fisheries unit, as well as very often U.S. Fish and Wildlife, will go out and do electrofishing surveys. Since eDNA has come online, we've been able to take samples of environmental DNA at the same time. For this first detection in the Hudson River in 2019, we just sort of did opportunistic sampling, meaning in areas that it was convenient for us to get them that also match snakehead habitat is where we take samples. But this did mean that it wasn't exactly, every 500 meters or anything like that. We didn't find anything in that particular situation, which was great. But that did get us ready for the next detection that we got in the Delaware in 2020. An angler caught a snakehead and did provide us with a photo. It's nice when anglers are able to provide us with photos because it, adds a little bit more credence to what they're saying. We can see from a photo whether or not they mistakenly identified the snakehead as a bowfin. In this case, they were correct and it was a snakehead. Once again, us regional fisheries staff and U.S. Fish and Wildlife went out and did surveys, especially focusing on the oxbows and mouths of tributaries where they're most likely to be, and where the angler said that they were fishing. At the same time, we did eDNA collection. We collected eDNA sort of in close proximity to where we were doing the electrofishing, because it's very possible that the environmental DNA is going to be able to capture fish that were in the area recently, but maybe aren't there anymore. And with this sampling method, we were able to get positive eDNA hits at the same sites that electrofishing was able to pull snakehead out of the water, which was great because that gave us a lot of confidence that the eDNA is picking up where the snakehead are. And we got negative hits elsewhere, which was also nice to know because this was showing us that this was probably just a couple of individuals in one place and not, potentially a massive population all throughout the river. We are a little uncertain right now what exactly the extent of northern snakehead in the Delaware River is, but we're working on some methods and some studies to kind of get a better idea of, exactly how far along the river they are and where exactly they are and potentially, how prolific that infestation is at this point. For the Bashakill WMA, we know that these fish are able to move great distances, as I said before. We once again got an angler that was able to catch a snakehead and they provided us with a photo of that snakehead. It's really great when anglers are able to do this for us because it really, provides us some point locations to start looking for these things. We went back out, this is old hat for us at this point. We know what we're doing. But this time we decided with the environmental DNA collection that we do 500-meter spacing. And this was done with Lower Hudson Prism, with U.S. Fish and Wildlife, and with the DEC. So it was, a multi-jurisdictional effort, a lot of people coming together to do these sorts of things. With this sampling method, so you can see here, all of these blue points are our sample locations. And that little white point kind of in that wider water body area is where the angler caught the snakehead. So we caught up and down stream of that location. And we weren't able to catch anything with electrofishing, but we did get positive hits for eDNA. There can be a lot of reasons for that. It can be that, maybe we were picking up DNA from the snakehead that was caught by the angler that was still floating around. It could be that there were smaller individuals that, weren't caught with electrofishing. It could be that electrofishing wasn't catching just exactly the right areas. But it was good for us to get that positive hit. We know that there are snakehead in the Bashakill and that it's probably not a large enough population for us to catch with e-fishing, especially in 2021. The nice thing about us doing the 500-meter spacing is that we are able to do replications of this unlike, you know, us doing the opportunistic sampling where it was just sort of wherever. We get a way better idea of where the snakehead are and where they are not. So this is our positive and negative detections. The negative detections are the little blue dots. The positive detections with the numbers inside show how many replicates were listed as positive. So the higher the number in that little yellow dot, the more certain we can be that there are snakehead in an area. So like 7s and 8s, that there were a lot of replicates, meaning that there's a higher chance that there was a snakehead in that area recently. And so you can kind of see where that angler caught that individual. There are some pretty high replicate positive detections. It was at this point that we really needed, you know, now we have some positive hits in the Delaware, we have some positive hits in the Bashakil. We were starting to get kind of concerned about the Hudson watershed. We really needed to understand if snakehead had made it into the Hudson already, or if we had time to do something before they did. And so with that kind of 500-meter spacing, we in 2022 went to delimit the distribution of snakehead in the Delaware Hudson Canal area, and the wetlands that are connecting them, which is all in that Bashakil WMA and the canal. And the connection in particular, so the water body, the Bash-Urkil, is the connection between the two water bodies. It's always interesting to talk about our sampling in the Bashakil and specifically in the Bash-Urkil. It gets a little bit confusing for everybody. But we did the eDNA sampling 500 meters apart. Again, a lot of people coming to help us. It was DEC, the Lower Hudson Prism, and U.S. Fish and Wildlife. And for our results that we got positive eDNA within the WMA, but upstream of the WMA, we did not get any positive hits. And this really, there we go. This really led us to believe that snakehead had not distributed into the Hudson side of the Delaware Hudson Canal, which was great, but meant that we probably should do something about that rather quickly. So that was spring 2022. This is fall 2022. And again, you can see that the little triangles are no detection, and then those little circles, depending on the color, are the number of replicates that got positive detection. So we see that there are some pretty high level positive detections just outside of that canal area, but that there are no positive detections within the canal itself, which is great. Slides, please. Okay, give me 1 second. Slight technical difficulties. Okay, let me play from current slide. So we went back and continued to do a lot more of this sampling and continued to find that there were no positive detections on the Hudson side of the WMA. Okay, it's just really going to struggle with these animations, I think, a bit. I might have to do this from now on. So we also wanted to capture reproduction of snakehead, and that was done by our inland fisheries team. Actually, I can go back because this is our 2023 sampling. Yeah, so in 2022, we found no individuals on the Hudson side. We went back in 2023. We continued this sampling. I love technology. We continued this sampling, and we continued to find that there were no replicated positive detections on the Hudson side. We did find 1 or 2 positive detections, kind of past where we were expecting, but we think that that was probably either a bird eating a fish and distributing its DNA sort of across into that area, or it was contamination on our part because we were never able to replicate those positive detections on the other side again. Let me find the slide I'm talking about right now. There we go. And so with these eDNA surveys, we went back and did the eDNA sampling monthly. And so we have a pretty good back catalog of the past couple of years of monthly eDNA detections, and mostly, thank goodness, non-detections within the Delaware-Hudson Canal area. While we were doing the sampling, we did get fairly consistent hits for northern snakehead in the northern WMA. But we, again, other than the 2 occasions that we got weak positives upstream of this, we really are not getting consistent positive hits upstream of the northern WMA, which is very positive for us. And again, gave us a chance to kind of do something about it while we were doing these samplings. Gonna chance getting a larger screen again. Our inland fisheries team went out and did Young of the Year sampling. There we go. They were able to capture quite a few Young of the Year. There were over 2,000 Young of the Year netted after an angler reported a location that he saw a snakehead. They were also able to capture the guarding adult, which was great as well. So you can see how little the snakehead are when they're first hatched. But again, they end up in these fry balls. So it can be like, to the trained eye, I definitely struggle to see fry balls when they're in the water, but for our fisheries team, they're really able to see, where it's likely that there are a bunch of young fish, head in that direction, and electroshock there. And then, if they catch a adult, then they know that the young are also snakehead. But they are very, very small when we're first catching them. And that was in 2022. Last year, in 2023, there were no fry balls that were detected or captured. We're thinking that this is likely because the snakehead, very fortunately for us, probably had a low reproduction year because of high water events, making the temperature a bit lower than is preferred for their reproduction, which you can see here. So all that flooding did have a positive effect for us, which is nice. Given that we felt at this point that we had a pretty good idea where the snakehead were within the Delaware-Hudson Canal area, we decided that we were going to try to recreate a separation between the Delaware and the Hudson. Just a reminder, because I know that this is one of those things where people see that we're building a barrier that fish can't get through, that the connection between the Delaware and the Hudson Canal was an artificially created connection. And so we're just really trying to reestablish the separation between these two systems. And so we're not too concerned about blocking the flow of fish. Except for the one that we are concerned about. We don't want them to head into the Hudson. So we installed a temporary barrier in 2022, which you can see right here. It's kind of just a little fence. It was installed by DEC Inland Fisheries team, so not contracted construction workers. So I think they did a pretty good job, all things considered, because we knew that the process for getting something more permanently installed was going to take some time, and it did. And we were able to improve the barrier. There it is. In November of 2023, this was a professionally installed, contracted barrier. It's got helical piles, which means there are those poles go pretty deep into the water. It should be fairly robust, and it's got 1/4-inch hardware mesh around the outside. So we're talking like 1/4-inch holes. So potentially those small fry could get through, but their guarding adult would not be able to make it through this hydrologic separation. So this is still what we're considering a temporary barrier. We are looking at more permanent barrier options and are still in the early planning stages of that, since anything more permanent is going to take a lot more permitting and probably a little bit of involvement from either Army Corps, since this is technically still considered a navigable waterway, as well as potentially DOT. So it will take some doing to get anything more permanent than this installed, but we are looking at that, both hydrologic study wise. We have the hydrologic study finished, and we know that if we were to install something more permanent, how that would have an impact on the surrounding area. But we're also working on things like design and permitting and cost and all those fun things that you have to worry about when you're doing a high level project like this. In the more recent times, we did some improvements on the towpath surrounding the one side that you can kind of see to your left there. Most of that little land area also tends to get a bit flooded, and so we were concerned about the flooding being able to reach around the barrier. we had seen some evidence that that might be possible. And so right now they're actually heightening that towpath in that area to prevent the flooding waters from being able to reach over it. So we're feeling fairly confident that this should stop snakehead, at least for the near future, until we can find a more permanent solution to the situation. Here's another photo. You can see that we're worried about buildup on either side, but we've kind of noticed that even this is after a high rain event, and the buildup of detritus on either side really isn't so bad, and that it held up pretty well even after we got a good amount of rain. Another thing that we're looking into is the potential impact of snakehead on the areas that they've already reached. And so in a 2022 fisheries study, these are all of the fish that were present within the Bashakil WMA. We've been continuing studies like this, and in fact, just this week, some of the folks from CRISP came out and helped some of the ESF folks with a community study to understand how snakehead could potentially having an impact on the food web in these areas. So that is something that we're looking into because, we want to prevent snakehead, but then we also want to understand the impacts that they're having in places that they already are at. So for our future plans, we're going to continue to monitor the temporary barrier to make sure that it's doing its job. We're also going to continue to do our, we've moved to every 6 weeks, going back and doing environmental DNA surveys. So far this season, we haven't gotten any positive detections, so that's really wonderful. We also have sort of an emergency detection plan where we take more samples than we actually analyze for the eDNA, but if any of those eDNA samples are positive, we have these backup samples to then test as well to get more fine resolution results. We are also doing some studies to understand the genetic connectivity of these different populations. We want to know if all of the New York introductions are a bunch of different introductions, or if it's possible that, one fish has moved to all these different locations and what that genetic connectivity looks like, because that's going to give us an idea of, how quickly and how far snakehead are really able to spread within this expanded range. And so that can give us an idea of how much time we potentially have from a detection to it reaching a different area. Again, we're looking at doing that we are currently doing the food web study in the Bashakil wetlands. And we're going to be doing some more robust eDNA surveys, both in the Delaware and the Susquehanna, to monitor the populations that we know are already there and understand exactly how robust those populations are. We're going to continue our rapid response and, regularly scheduled surveys. So if an angler sends us photos and stuff, we, activate our rapid response protocol. But then there are also places where inland fisheries just goes out and does routine surveys, and those locations rotate every year. We also have education and outreach opportunities. We have a northern snakehead fact sheet that we circulate, and then we're also beginning to target some cultural and religious communities to try to encourage them to, understand that releasing of certain species is very detrimental to the environment. So we're trying to reach them and help them understand that that there could be better alternatives to this cultural practice, instead of releasing snakehead into our waters. And so again, this was a pretty, massive project that was conducted by a bunch of different groups, again, including some people from CRISP and the other prisms, and Stephen Pearson has done so much of this work and really leads the DEC side team of these things, as well as a bunch of our inland fisheries folks. And I'm just really happy that I get to be a part of it as the Region 3 coordinator. And I'll thank everybody. That's my full talk. And if anybody has any questions, I'd be happy to answer them.
Thank you, Ashley. Thank you for overcoming the technology and making this possible.
It's always been a pleasure.
Do people have questions?
Is it legal to release them? Are they, no.
No,
Is there
so they
any
are regulated. Yeah, they are regulated under Part 575 as a prohibited species to possess or to sell or to release. That doesn't stop people But they are a regulated species. And so, if our environmental police go out and they find them being sold, they can, repossess those fish and give those people tickets and stuff like that. So.
The chemical treatment you mentioned.
It was a rotenone treatment. Which is why it's not exactly feasible for places like the Bashik Hill that have a fairly diverse and robust fish community. We'd really prefer to not, because it's non-selective. The area that we were able to do that at, it was a fairly small enclosed pond. They were able to remove a lot of the desirable species, do the treatment, and then put everybody back. But it's not really possible to do in flowing water, and especially in larger areas. There are potentially detrimental effects. We've been looking at maybe trying to block off parts of the Bashik Hill if it came to that, to do a rotenone treatment if it came to that. But, we know that it's a bit complicated and we'd really prefer to not have to do that.
A question. How good is the signage around the Bashik Hill to encourage the fishermen not to catch and release?
There is some signage in areas that we know that there are populations and that there are fishing spots. The part of the Bashik Hill that we have the barrier, we have a sign on the barrier explaining what's going on here. But we don't put too much signage in the area because we're actually not encouraging people to go there. It's not an overly used footpath because it's flooded, honestly, most of the time. And we were more concerned with people we didn't want to draw attention to the area and the problem in that particular location. But at most of the places where we know that people launch boats and fish, we have signage. I don't know if it's enough. If you have locations that you're thinking about that need a sign, you can definitely reach out and we can look at that location and see if we can put a sign in there for sure.
Thank you.
Any questions?
Stop the recording.
Thank you, Ashley.
Great.
We don't seem to have any other questions. It was a great
Awesome.
presentation.
Thanks. Yeah, if anybody has any other questions or whatever, definitely feel free to email me, and I can provide you with as much information as we have.
Thank you.
Thank you.
And we'll probably at some point, if anything changes, you're always welcome to come to a partners meeting and give an update. So yeah, just let us know.
Great. Awesome. Yeah.
Keep us
sure.
updated.
Yes.
Thank you,
definitely.
Ashley.
Yep. Thanks so much.
So we were going around the room with